So what I'm going to do is I'm going to show you how to get to the lab, and then I'll open the lab and I'll kind of like walk you guys through it. So if you haven't done the bacteria lab or you're kind of working on it, if you want, you can you know, follow along and you know, collect the data with me, and then you'll kind of have that out of the way. One less thing that you have to do, since I know you guys have a lot going on in this class. So here's the course. I have it on student view, so this is what it should look like when you guys see it. These buttons here go to the like course resource newsletters, where I've got like tips and hints and stuff like that for the different modules. So this one's in the bacteria and viruses web, so I'm just going to click on that, and it's going to take me to this. And you can see if you need a PDF of the lab, it's right here. You can see there's different information, different things that I've done where I've discussed content, stuff like that. Kind of a study guide I made for my kids who are in the face-to-face classroom. So there's all that stuff in here. So we're going to do the virtual bacteria lab. So you can either go to page 5 of the module, or I've made it really easy. You can just click on the handout. Now, this link on the handout does not work. You see how it says .swf? That doesn't actually work. If you go to this lab that I've put up here and download that one actually has a correct link. You can see the .html at the bottom. So definitely check that out. You know, at least get that link. So let me go ahead and open that lab. Okay. Here's what I'm gonna do. I think I opened it in Word the other day, and it let me edit it. Okay. Yeah. Excellent. Okay. So can you guys see like the document I have up, the Word document? Hopefully, yes. Okay. Well, I'm going to hope that you can, since I'm not hearing from y'all. And so what I'm going to do is I'm going to take this link right here, and I'm just going to copy it and paste it into a brand new window. Okay, and this is going to take me to this lab. Now, the first time that you open it, it may ask you to, like, do you want to run Adobe Flash or whatever, and you'll say yes. Okay, so let's see what we've got going on. So it wants you to read this information right here. This has a lot of information about bacteria. This information will help you answer the questions at the end of the lab. So it's pretty important. And then what you're going to do is you're going to choose 2 of these bacteria. So we have these 3 bacteria here, and you're going to choose 2 different ones. It doesn't matter which 2. Okay? And then you want to write a hypothesis that says which substance will be the most effective at killing them, and which will be the least effective. Okay? So you could say, like, if species of bacteria, depending on whichever one you choose, is exposed to whatever substance that you choose, most will die, or something like that. If species of bacteria is exposed to substance that you choose, very few will die, or something like that. That's not really an excellent hypothesis, but you know, it's off the top of my head, so we're going to go with it. And so now let's go here. So here we're going to pick a bacteria. So I'm just going to do the first 2. Okay. So these are the 2 bacteria that I'm going to choose. Okay. So now that we've got that part, we need to go back to the website, and it's going to give us the procedure. Okay. So it says, inoculate the agar in the Petri dish by clicking one of the test tubes. So I'm going to take this, and I'm inoculating the agar with the Staphylococcus, okay? And it says it right here in case you forget which one you used. Okay, and then it says we need to put these paper discs, and it's really important that you put the paper disc like right where you want it, and you don't like pick it up and move it around, because that will cause some cross-contamination. Okay, so there I go. Okay, now I'm going to click on this little incubator oven thing, and it's going to put my bacteria in the oven. And I'm going to turn it on, and then I'm going to stop it when it hits 24 hours. Okay, so now we have our different ones, right? And we're going to see, we have to measure it. So this one, I don't know. I mean, it looks like there's not even really any bacteria. It's just a little circle. So what is this? 1, 2, 3, like, yeah, like 3.5 millimeters. So I'm going to put that there. And again, you guys can follow along with your lab, so then you don't have, you know, you've kind of got it mostly done when you finish this session. That one's like 14. And it's really important. Do you see how I'm not going at the edge of the ruler? I'm starting at the 0 millimeters. That's really important. Otherwise, we're not going to get an accurate measurement. So this one looks like it's 30 exactly. Looks like it's about 20 exactly. So 10. 12. And you see also I'm trying to measure it at the fattest part of the circle. I'm trying to get that diameter of the circle. Okay? So we've measured this. And then we're going to hit the reset button. And we're going to try our next species. Okay, so I'm going to do this Haemophilus. And I'm going to do the same thing. 1. Okay. I'm going to hit the oven. I'm going to put it in the oven for 24 hours. Okay. So here we are again. That sterile paper is again like 3.5 millimeters. This one was about 17. So you can already see, like, the antibacterial soap is much better, a little better at least, at killing the Staphylococcus than the Haemophilus. Here we have, what, 31? Bleach doesn't really do anything to those guys. You know, so when you're cleaning your house, so 21. Disinfectant didn't do much either, did it? 29. This guy's like really much less than the penicillin, right? The penicillin did a pretty decent on the other one and not really anything on this one. Here's 27. Same with the amoxicillin. Now, unfortunately, you know, amoxicillin is one of those antibiotics that they usually give people first because it's cheap and it's, you know, it is usually pretty widely effective. And so, but, you know, sometimes it doesn't work, and then you've got to kind of start over. So here's the data that I collected, okay, and then you're going to want to answer these questions. Okay, so for each species of bacteria, state which substance caused the most and the largest zone of inhibition. Okay, so the zone of inhibition would be like, it's like more likely to kill them. Okay, so we could do S. Most inhibition. Least inhibition. So this would be the one, the most inhibition, which would be like the smallest circle. This would be the smallest measurement. And this would be the largest measurement. Okay. And then you could do this for the second species of bacteria as well. Okay, did your data support your hypothesis? You know, which thing did you have up here, right? Did you have bleach, or did you have, you know, what did you have, an antibiotic, penicillin? So you would just say, you know, is your hypothesis supported or not? What was the purpose of the sterile paper, disc 1, in the experiment, right? That sterile paper was basically nothing, right? So what is it when it's nothing? It's a control, right? It was a control just to see what would happen. If you didn't have anything. Distinguish between how each antibiotic kills bacteria and how the chemical agents in the experiment kill the bacteria. Okay, so what's killing the bacteria, what's not, which bacteria dying more. If you were a doctor and saw 2 patients, each one infected with your chosen 2 bacteria, which antibiotic would you recommend? Right, so these are the 3 basic antibiotics that we had here. Penicillin, amoxicillin, and erythromycin. Okay, so one is much better at killing others. For example, penicillin and amoxicillin were pretty good at killing the staphylococcus, but hardly did anything to the hemophilus. And so if you had hemophilus, you'd want erythromycin. That's why a lot of times if they suspect you have a bacterial infection, they'll take a culture because they want to see what kind of bacteria you have, because not every antibiotic is useful for every bacteria. And then it says, what are some limitations to testing the effectiveness of antibiotics in this way? If it involved a real person, what would give you more confidence in your results? So you could say, what you'd prefer for a real person. And then this is basically showing you what you're going to get, where the most of your points come from the lab. So you can see 60 of your points, so 60% of your lab is answering these 6 questions. So it's important that you kind of think about those and you do them, you answer them fully. So that's kind of just how you kind of go through and do this little lab. Hopefully it was helpful. Again, the link to the lab, you can go to the link right here. I've got the working link here, or you can download this PDF and it will also have the working link. Okay, so either one of those will get you to the lab, the correct lab.